Hi guys, I'm Mrs. Elias for those of you who haven't had me in class. This week's tutorial video is over protein synthesis and we'll talk about mutations a little bit. The first thing that I want to do is go over some review stuff. There's the basics of protein synthesis and then we'll practice doing the steps a little bit. But first thing to remember is that synthesis means to make or create. And so protein synthesis is the process of making proteins. And proteins are a biomolecule. They are a very broad category of biomolecule. They do all the things in our body. So for example, you have transport proteins in your cell membrane, and you have enzymes that break down or create a bunch of things, anything in your cells. So they do all the functions. There's so many proteins and they're all coded for by your DNA. So the first step of protein synthesis is transcription, taking those sections of DNA, which are called genes, that code for a protein, and you have to transcribe them. The first step is transcription, turning that mRNA template with the gene sequence into a strand of mRNA. And this happens in the nucleus because that's where DNA is. And if you were to remember, DNA is too large to leave the nucleus. And so we have to turn the DNA template into mRNA, and this is done by enzymes, RNA polymerase. But if you have a gene sequence like this, the other side of your DNA strand would be the complementary sequence. Okay, but only one side is our template. So then RNA polymerase, which just means it's the enzyme that builds RNA molecules, RNA polymers, unzips the DNA and makes this bubble, or this, it's not called replication bubble, but makes this bubble kind of like DNA replication does. And then it builds the complementary strand of RNA by using base pairing rules. Now, if we remember, the main differences between RNA and DNA is that RNA is single-stranded and it has uracil or U instead of thymine, which DNA has. Those are the main differences. And then the sugar, ribose sugar instead of deoxyribose sugar. But RNA polymerase builds this complementary strand of mRNA, and then this DNA bubble is going to zip back up because the mRNA is going to leave and go to a ribosome. This is transcription though. So RNA polymerase builds the complementary strand of RNA using base pairing rules. And so they're different because it's RNA. A bonds with U because there is no T in RNA. T always bonds with A. C bonds with G. G bonds with C. A bonds with U. C bonds with G. So that's how we got this mRNA sequence. So after transcription, the next step is translation. Translation, so our mRNA strand that we just created in transcription was made in the nucleus, transcribed in the nucleus by enzyme RNA polymerase. It leaves the nucleus and goes to find a ribosome, and ribosomes are the structures in cells that build proteins, or where the final step of building proteins is. So those mRNA messages from transcription get taken to an ribosome, and we're going to translate that mRNA into amino acids. And this is done by tRNA molecules, which match up with our mRNA sequence and then leave the correct ribosome, amino acid in the sequence, because just like specific sequences of A's, T's, C's, and G's, or those nitrogen bases, code for genes, having a correct sequence or a specific sequence of amino acids builds specific proteins. So let's do an example of this. So if our mRNA sequence, and remember mRNA stands for messenger RNA, if our mRNA sequence is something like this, there can't be a T. Okay, we'll see what that gives us. But if you have an mRNA sequence, let's say it's this. We read or translate mRNA in threes, or the nitrogen bases in 3 at a time, which is called a codon. Each of these is called a codon because they're part of the mRNA strand or mRNA sequence. So right here with these 12 nitrogen bases of our mRNA strand, we have 4 codons. And each of these codons is going to translate into an amino acid. So our tRNA molecules match up with our mRNA codons. And these tRNA molecules are made out of an anticodon, and then the tRNA molecule, and then on the other end, they have an amino acid that they're going to drop off. This one happens to be methionine. I just know that because I know what this gives me. But this part of a tRNA molecule is called an anticodon. So each anticodon of a tRNA molecule codes for or matches up with a codon, telling it, that's how it knows which amino acid to leave. That's how this sequence gets translated into amino acids in your cells. Please excuse my terrible tRNA molecules. They keep getting worse. Okay, so tRNA molecules with their anticodons match up to the mRNA codons, dropping off a specific amino acid in sequence. These form bonds between the amino acids, which are called polypeptide bonds. You don't necessarily need to know that. But once you start bonding amino acids together in sequence, this whole thing becomes a protein after it folds up a couple. There's a couple more steps to fully being a protein, but what you guys need to know is that a sequence of amino acid, an amino acid chain, or a polypeptide chain is another word for it, is a protein. So this is the last step. We went from transcription, we made our mRNA from our DNA template, and then we're translating it using anticodons to leave the correct amino acid. Now, how do we know what the amino acids are? In your cells, the correct amino acid is just attached to that anticodon tRNA molecule. That's how it knows what protein or what amino acid to leave. But for us, like on a STAR test question, which is why we're here, you would use what's called a codon chart. So let me share one of those with you real quick. Here we go. So you have a codon chart. So just a reminder that you start with your first base, which, for example, how I got methionine from AUG. Now, thing to remember is that we're using the mRNA. You don't have to ever, on a test question, go from mRNA to tRNA. You just go from mRNA to the codon chart. Basically, scientists have skipped that part by creating a codon chart. So we start with our first mRNA base was AUG. So you start with the first base A, which gives us this row somewhere here. Second base is U, so we've narrowed it down to this box here. And then G is our last base, so we know it's the last one in this box. So it's methionine. Now let's do AUC, our next codon. So again, we start with A, and again, we start with U. But because C is our last letter, isoleucine is the correct amino acid in this order. And then our next codon was CCA. So let's look here. C, first base, it's in this row. C, second base, it's in this box. And then A, which if you notice, all of these are the same amino acid. This is possible because there are 64 possible codons and there are only 20 different amino acids. So sometimes different codons give you the same amino acid. So CC anything gives us proline. Then our last one was AUG. UU. So A, again, it's in this row. U, it's in this box. And then U is the top one, it's isoleucine again. Okay. So let's do a couple practice questions over transcription, translation, protein synthesis. So looking at this practice star test question, you see you have an mRNA strand and you have these little T-shaped, these are very T-shaped molecules, which are more than likely representing tRNA because we see here an anticodon, which is matching up with this mRNA codon, and then it's leaving amino acids and they're starting to form a chain. So the purpose of this process, we know that we have mRNA, tRNA, and amino acids. So that tells you it's translation. So what's the process of translation? The purpose is to translate our genetic code into a specific sequence of amino acids. It's not to replicate DNA, that's a different process. There's no RNA or amino acids in the process of DNA replication. It's not assembling nucleotides in an mRNA chain. This is transcription, what that's describing. That's the first step of protein synthesis. And then we're not making amino acids. We're using already produced amino acids because you actually get those from digestion and stuff, digesting food. To translate the genetic code into a sequence of amino acids. So the correct answer here is J, hopefully by process of elimination and recognizing that if there's an mRNA strand, tRNA molecules matching it up and then leaving amino acids, you know that's transcription. Here's another one. So the initial step in gene expression, which is just another way of saying protein synthesis, then gene expression is after those proteins get made, they express your genes. Like the reason you have blue eyes or brown eyes is because of the proteins that were made in protein synthesis. So we take our double-stranded DNA, it's unzipped. As you can see here, we have that bubble that's created. And because we have a single-stranded molecule being created and leaving this bubble, we know this is transcription. If it were DNA replication, both sides would be replicating. So that's how you know it's not DNA replication. So which process and product are represented? So we have the process we see here going from double-stranded DNA, opening up and creating single-stranded, we can assume it's RNA. So we have the process of transcription is what this is called, and then the product is mRNA. Process of elimination, we know it's not a protein. We don't see any amino acids in this picture. It's not tRNA, but also replication doesn't make tRNA molecules. And recombination and polymerase. Polymerase is an enzyme. There's no enzymes here. Recombination, I guess that's some kind of mutation thing. So that it's not an option. If this were translation, it would look more like this picture. This is translation. This is transcription. Okay. So this question, which of the following polypeptides, meaning protein, polypeptide is another word for protein, is coded for by the mRNA sequence. So we have our mRNA sequence here. The 5 and the 3 just mean that this is the direction. We read from left to right. It just means this is the start, this is the finish. You don't necessarily need to know that, but anyway, it has to do with the direction of the sugars. Don't worry about it. Just always read from left to right like we always would. AUG is our first codon, because remember you read the genetic code 3 letters or 3 bases at a time. AUG, so if you go here and do A, it's in this row. U, it's in this box. And then G, it's methionine. So that narrows it down to G and H because it has to start with methionine. Now we really just need to figure out what the next one is. So like test taking strategy, if you don't need to translate the entire thing, don't translate the entire thing. We already eliminated F and J. So methionine or methionine. So now we need to see if the second codon gives us ASP or valine or asparagine or valine. So GUU is the next codon in our sequence. So we start with G as our first base. So we know it's in this row. U is our second base, so we narrowed it down to this box, GU, and then U is the top one. But again, the entire box is valine, so the answer to this one is H, because it's not asparagine. So we eliminated everything but this one. And if you wanted to just verify, then I guess you could translate the entire thing, but you don't really need to. It's up to you. Of course, you do have 4 hours to take the STAR test, so there's plenty of time if you want to translate the entire thing. Okay, we'll do this one real quick and just discuss gene expression a little bit. Human bone, muscle, and nerve cells all contain the same number of chromosomes with the same complement of genes. What enables these cells to perform specialized activities? The ability of some cells to remove unnecessary DNA sequences. That's not possible. I definitely said this in my class. I don't know about Mr. Cotton and Mr. Zamora, but a lot of your DNA is what we call junk DNA. It actually doesn't code for anything. So your cells don't take out any DNA that they don't need. All of your cells have all of your DNA. The regulation of gene expression within each cell type. This is the correct answer because gene expression, meaning which traits your cells are using basically or expressing, it's different within your skin cells. Your skin cells use different genes and different traits than your blood cells. Otherwise, all of our cells would look and act exactly the same. This is possible because there's enzymes, obviously, and proteins in our body, in our cells that control which genes get turned on in specific cells. And this is called cell differentiation, and it's because of gene expression. So the fact that our cells do different things is because of regulation of which genes are turned on and which genes are turned off. It's why your fingernails are only produced at the tips of your fingers and your toes, for example, and not all over all of your skin, which is, thank goodness for that. And it's why red blood cells look and do the job of carrying oxygen in our red blood cells instead of skin cells, which definitely don't do that. So, and brain cells, it's the difference in all of your cells. This is not caused by mutation of genes because hopefully all of our DNA and all of our cells is the same. We know that mutations do happen during cell division, but for the most part, 99.99% of it is the same. And just which one's turned on and turned off in specific cells, and that's controlled by gene regulation. The use of different methods of cell division. There's only 2 kinds of cell division, mitosis and meiosis. Sorry, my fingers. Mitosis makes all of your somatic cells anything except a sperm or an egg. So that's not what makes our blood cells different from our brain cells, different from our skin cells. So that is G, the regulation of gene expression in each cell type. Okay, last thing I want to talk about in this video is mutations. So let's go back up here real quick. So there's different types of mutations which occur in DNA, and I know I mentioned that mutations happen random. Every gene mutation is random. Every time your cells go through mitosis, there's about an average of 3 gene mutations. There's different ways of mutating DNA. For example, you could have an insertion. So this is adding 1 or more extra nucleotides when DNA is replicating. You could have a deletion, deleting 1 or more nucleotides. A substitution is where you simply change one base for a different base. And then a point mutation is the same thing as a substitution, really. It's just changing one thing. It doesn't make your DNA strand longer or shorter because we're just switching one thing out. And then a silent mutation is one that doesn't change the amino acid sequence. And that's possible because when you do transcription and translation like we did, some codons code for the same amino acid. And so if you're not changing the amino acid, you're not actually changing the protein. So let's go back down here. There's one practice question over this. So different types of mutations can occur in DNA. The diagram represents a type of mutation. So here we're looking at, this is actually a chromosome and zoomed in on the gene sequence. Chromosome zoomed in on the gene sequence in the DNA strand. Which statement describes the mutation in the diagram? So you have to compare these two sequences of DNA, GAC, TAG, CAC versus GAC, TAA, GCAC. If you count these, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 bases. And over here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 bases. So just counting alone, we know that we added an extra base here, right? This would not be a silent mutation, more than likely, because insertions and deletions add or subtract from the entire length of your DNA sequence. And they're what we call frame shift mutations, and they change everything before, or not before the mutation, behind the mutation. And here, let me kind of show you that real quick. So using this mRNA, which we can just assume my DNA is what coded for it. So if we mutate the DNA, say our original DNA sequence would have had to be TAC, TAG, okay, this DNA gives me this mRNA using base pairing rules, okay? But if we inserted a base here, let's say a C, then that would insert an mRNA codon also. So if we had a G mutation in our DNA, it also affects our RNA, because this would be a G now. And so then when we read our codons, it would be 3 bases, 3 bases, 3 bases, and then these 3 bases, leaving this guy out. He doesn't mean anything. It's just shifted everything down. This is called a frame shift mutation, when we actually shift. If there was more bases behind this, which a sequence of mRNA would be way more bases, all of it shifts down 1 because we inserted 1. And so GAU is going to give us a different protein than AUU. And so insertion almost always changes the amino acid sequence by a lot. Same thing with deletion. If we deleted a base in our mutation, like let's say we deleted the A because the T was gone. So now we have to read this as this. And then these guys don't code for anything because you have to have 3 codons to translate into an amino acid. Go back. All right, so it's not silent. Okay, and silent mutations never happen because of a different amino acid. It's because the mutation gives the same amino acid, meaning it doesn't change the protein. This one is just saying that it does change the protein. It's not a substitution because we didn't just switch a letter over here for an A. We actually added an extra A. We didn't delete anything. We kept all of our C's, our cytosines. The right answer is a base is inserted into one strand of the DNA. So we added that extra A or adenine into this strand of DNA, making it longer. Okay, that's the last practice question. So I covered everything that I needed to. I hope this was helpful. Make sure you go and complete the quiz that goes with this video so you can get your extra credit points. Thanks, guys. Bye.